Hello everyone, my name is AJ Keefe, and in this presentation I'll be reviewing the function of centrioles, as well as the modulation of GABARAP. It's a cargo adapter receptor on autophagosomes. I'll use this paper as an example of how these concepts are being currently researched. The paper is titled Centriolar Satellites Control GABARAP Ubiquitination and GABARAP Mediated Autophagy, published in the Journal of Current Biology. I want to extend a special thanks to the authors for their contribution to this field of research. And for this video, I recommend my viewers pause the video at each new slide and read through the material and analyze the graphics before playing my commentary. And if you have any questions, please comment below. The authors, based on previous research, were interested in a potential relationship between the centriolar organelle that we see up here and the process of autophagy that we see down here. So the centriole is also called the microtubule organizing center, and that's for good reason, because it's the site of microtubule biogenesis or nucleation. So microtubules are distinctly different from something like actin or intermediate filaments, because they cannot nucleate wherever they want, and instead all the microtubules in a cell can be traced back to the centriole right here. And so any given cell can have dozens of microtubules that are sprouting from its centriole, where they are constantly undergoing this process of search and capture. These microtubules will grow, they will search for targets, and if they don't find a target, they collapse in what's called catastrophe. So microtubules are kind of flailing around the cell, constantly growing and collapsing until their plus end binds some kind of stabilizing protein on something like the plasma membrane or a mitochondrion. So all microtubules can be traced back to the centriole, and immediately surrounding the centriole is this amorphous mass of protein called the pericentriolar material. It's basically like a protein jelly that roughly defines the centriolar organelle. And this protein called PCM1, we see it right here. PCM1 is thought to organize and recruit proteins to the centriole, and kind of defines the organization and the makeup of the pericentriolar material. And so PCM1 would actually be all throughout this pericentriolar material. And another interesting aspect or characteristic of the pericentriolar material is that it's not necessarily confined to the centrioles, and it can actually be trafficked up and down the microtubules in what's called these centriolar satellites. So these centriolar satellites are moving up and down the microtubules, which, remember, they always provide a path back to the centriole. So when a couple of centriolar proteins link up with dynein, they can actually travel out of the centriole, and they form these centriolar satellites. And they're thought to be nucleated, again, by PCM1. So PCM1 is essentially a marker for centriolar satellites that have migrated out of the centriole. And another interesting thing about centrioles that I'm not going to get into, but I just wanted to briefly mention, is that they're actually the site of cilia biogenesis. And cilia are really critical signaling organelles in many different cells. And so when we're talking about centrioles, that's just something to kind of keep in mind. Anyways, the other main idea for this paper is autophagy, that we're going to be looking at this graphic down here. So autophagy is simply the catabolic process of self-digestion and turnover of cellular components. And in order for an autophagosome to recognize its substrate, it needs to be decorated with these receptors. Right here are the receptors. These receptors are of the LC3 family of proteins, and it includes GABARAP. Right here, GABARAP. So GABARAP is a protein that is cleaved by ATG4, and then it's lipidated onto autophagosomes by this complex ATG12,5,16. So this 12,5,16 complex simply takes cleaved GABARAP. So this is GABARAP that's already been processed, and it attaches it to the autophagosome by conjugating it to phosphoethanolamine. And so ATG-12-516 complex is just conjugating GABARAP onto phosphoethanolamine, which then anchors it to the autophagosome. And this ATG-12-516 complex is itself recruited to autophagosomes by the WIPI and DFCP proteins. So in this paper, wherever you see WIPI or DFCP, that means that it's initiating the recruitment of ATG-12-516, which will then conjugate GABARAP with phosphoethanolamine on the autophagosome's membrane. And so I don't want to get into too many specifics because I have an entire lecture on autophagy that you can go check out. But for this paper, all you need to know is that DFCP and WIPI signal the formation of the autophagosome. DFCP and WIPI recruit ATG-12-516, which takes cleaved GABARAP and attaches GABARAP onto the membrane of the autophagosome. You can see right here. So the next question is, why is this important? Why does GABARAP need to be attached to the autophagosome's membrane? What's the purpose of that? Well, GABARAP and LC3 are actually receptors. They bind to a very particular domain called LIR, L-I-R, or LC3 interacting domain on other proteins. So GABARAP binds to other proteins' LIR domains. What proteins contain these LIR domains? Cargo adapters. So GABARAP binds to cargo adapters through their LIR domains. And cargo adapters are bridges between autophagosomes and ubiquitinated substrate. So a cargo adapter, something like P62 or BNIP is another one. There are many different cargo adapters, but they contain this LIR domain, which binds to GABARAP or LC3. And these cargo adapters also contain a ubiquitin binding domain. So you end up with this kind of confusing looking complex where you have you can see it down here, you have a substrate right here. It's called cargo in this. So you have some kind of ubiquitinated substrate. It could be, aggregated protein or something. And it's bound to ubiquitin, right? It gets polyubiquitinated. This cargo gets polyubiquitinated. Then it binds to this receptor, which in our case is going to be something like P62. This is our cargo adapter. The receptor is a cargo adapter. And then what recognizes the cargo adapter is our GABARAP receptor. So we have GABARAP, which binds to P62, a cargo adapter, which binds to ubiquitin on the cargo or on the ubiquitinated substrate. So in other words, GABARAP indirectly binds to ubiquitinated autophagic substrate through cargo adapter intermediate proteins, which bridge autophagosomes with polyubiquitinated substrate. And we'll see in the next slide and in this paper that GABARAP, the autophagosome's cargo adapter receptor, has a special relationship with these centrioles and their satellite projections. So again, remember that PCM1 is a marker of the centriolar satellites. So let's get into the specifics. So this protein GABARAP, again, is important for autophagic cargo recognition and subsequent autophagosome nucleation, was previously discovered by these same researchers to bind to PCM1. PCM1, we see it right here. So PCM1 then causes the localization of GABARAP to the pericentriolar material. So the central question to this paper is, what significance does GABARAP localization to the centriole have? Is there any kind of special relationship? Does it actually mean anything? So the authors began by looking at potential LIR domains on PCM1 because GABARAP and other LC3 family of receptors are known to bind to their cognate ligands at LIR domains. So in other words, they already know that GABARAP and LC3 bind to Lear domains. So if PCM1 contained a Lear domain, that would make sense, because that would be how GABARAP would be binding to PCM1. So it turns out that PCM1 does indeed contain a Lear domain, and it's also required in order to bind to GABARAP. And this Lear domain on PCM1 localizes GABARAP to the pericentriolar material. And it turns out that during autophagy, this PCM1 and GABARAP complex, so right here we see during starvation, we see the this PCM1 GABARAP complex migrating out towards the plus end of microtubules. So this is a satellite that's migrating out of the pericentriolar material. And it essentially localizes to sites of autophagy marked by the presence of WIPI and DFCP. So WIPI and DFCP are coding this phagophore that then recruit proteins like ATG12.5.16, which lipidates GABARAP onto the membrane. So the presence of PCM1 at these locations right here suggests that the GABARAP PCM1 complex moved there as a centriolar satellite along microtubules. So these centriolar satellites are highly dynamic, constantly crawling up and down microtubules, and since they have a role, they may play a role in delivering GABARAP to sites of autophagic nucleation. So they are, in a sense, responding to metabolic stress. Furthermore, the depletion of PCM1, so they look at what happens when PCM1 is lost. And what they find is that there's a premature release and premature activation of GABARAP, and thus it causes autophagy. But this activation is kind of short-lived, because permanent PCM1 knockouts actually cause GABARAP downregulation. So PCM1 essentially sequesters GABARAP until it's needed for autophagy. And when PCM1 is lost, GABARAP is degraded by this pericentriolar ubiquitinase, MIB1. So MIB1 is, again, a ubiquitin ligase, and it causes the accumulation of immature autophagosomes and accumulation of cargo receptors. So when PCM1 is lost, it causes the accumulation of immature autophagosomes and accumulation of cargo adapters. So autophagy is basically stunted when PCM1 is lost, because it leads to GABARAP instability and ubiquitination and breakdown. So the authors theorized that PCM1 is essentially modulating the transport of GABARAP via these centriolar satellites, and it functions to basically deliver GABARAP to sites of autophagosome biogenesis. So this kind of elucidates a role and a function of centrioles in the modulation of autophagy. So centrioles may be a kind of autophagic organelle. In order to identify the nature of this interaction between PCM1 and GABARAP, they first identified this putative Lear domain right here, FVKV. So they then systematically mutated all of the nearest 10 amino acids to every other amino acid. And they quantified PCM1's ability to pull down GABARAP. So it's a really impressive and high throughput assessment of PCM1's interaction with GABARAP. And you can see the lack of pull down following mutation of these 4 amino acids, indicating a functional Lear domain, is right through here. So the lack of black wells indicates a lack of pull down when these amino acids are mutated. And in this bottom figure down here, we see a more straightforward immunoprecipitation of various LC3 proteins, including, most importantly, the 3 GABARAP isoforms. And PCM1 is pulled down by all of the GABARAP isoforms, as well as the LC3C. So you see there, I can't necessarily put my You see PCM1 is staining at the LC3 well, the GABARAP well, the GABARAP L1 and L2. These are different isoforms of GABARAP. We basically see when GABARAP is pulled down, these different isoforms are pulled down, PCM1 staining is positive in these wells, indicating a interaction between these two proteins. So these immunocytochemistry images are from fixed human embryonic kidney cells, and they're stained for PCM1 in green and GABARAP in red. And a simple assessment of colocalization can be observed between both proteins in these merged images. We see these close-ups, we see some colocalization, especially in this figure down here. And although somewhat disappointing, is the Pearson correlation coefficient is only 0.2, which is a fairly weak correlation, but it's a correlation nonetheless. And so there is at least some kind of interaction between GABARAP and the centriolar satellite PCM1. This is just some more immunocytochemistry of fixed hex cells. And we see the marker of centrioles, is this SSX2IP, is colocalizing very well with both PCM1 and GABARAP. We see lots of colocalization in the merged. So the colocalization between SSX2IP and PCM1 isn't too surprising, but the colocalization additionally with GABARAP is surprising. And they also look at pericentrin, which is another marker of the pericentriolar material, as well as I think some satellites as well. But basically, again, we're seeing that GABARAP not only interacts with PCM1 in various molecular assays, but it's also located at many of the same locations in vivo. So what's the functional importance of an autophagic protein, GABARAP, colocalizing with a centriolar protein, PCM1? Since GABARAP is recruited to autophagosomes during starvation to nucleate the membrane, they wondered if PCM1 also moves into the forming autophagosomes location as well. And it turns out that it does. So they labeled forming autophagosomes with DFCP down here, or WIPI. These, again, are the two membrane binding proteins that nucleate the autophagosomes membrane. And since they then found that PCM1 also localizes with GABARAP at these sites of autophagosome formation. We see pretty robust colocalization, especially right here. You see in red, you can see PCM1, and in green, you can see the forming autophagosomes. We see a lot of the forming autophagosomes have robust colocalization with PCM1. Again, that's a centriolar protein that's being recruited to forming autophagosomes. So we can say that GABARAP is recruited to autophagosomes along these centriolar satellite complexes, because again, PCM1 is a marker of centriolar satellites. So they posited that maybe the centriolar satellites were recruiting proteins to the forming autophagosomes through this PCM GABARAP interaction. And in the bottom right, we see a really important immunoisolation experiment. So this is basically an immunoprecipitation of certain membranes. And in this case, they immunoprecipitated the DFCP positive membranes. So they're pulling down membranes that are positive for DFCP. And so in other words, they're pulling down autophagosomes. And they stained for these different proteins, showing importantly that PCM1 and other centriolar protein, MIB, are co-immunoisolated with autophagosomes. And so I think this is probably one of the most important experiments of this entire paper, is that this immunoisolation of autophagosome membranes shows pull down of PCM1 and MIB1. And we also see, not surprisingly, GABARAP being pulled down as well. So this collectively means that PCM1 is being recruited to and binding and perhaps functioning in some way of promoting autophagisome nucleation. These sets of graphs are quantifications of immunocytochemical imaging experiments that are quantifying the number of spots per cell. So in this graph, they're looking at GABARAP, WIPI, double positive puncta. In this graph, they're looking at LC3, P62, GABARAP, WIPI. So looking at all these different stainings, and they're looking at the number of spots per cell in these two different conditions, ES and EB. So ES is a simple and straightforward nutrient deprivation solution. It's just a balanced salt solution without nutrients, and it just induces autophagy. And EB is that same salt solution plus bifidobacillin. Bifidobacillin blocks the acidification of autophagosomes, and it prevents their degradation through lysosome maturation. So for simplicity, just think of EB as inducing cellular congestion and induces the accumulation of autophagic material. And so in this top right graph, we see that GABARAP and WIPI2 colocalization increases following EB administration, which makes sense because when you block the degradation of autophagosomes, you see increased GABARAP WIPI2 colocalization. And what's interesting is that this upregulation is prevented by PCM1 mutations. So these are just various PCM1 mutations. I would ignore these E, these are phosphomimetics, but this S372A mutation on PCM1 prevents a critical phosphorylation site. And preventing PCM1 phosphorylation prevents the upregulation of GABARAP WIPI2 staining following autophagy congestion. So I thought this was really fascinating because PCM1 is a centriolar protein, and mutating this single residue on a centriolar protein prevents the autophagy accumulation following EB administration. In the bottom right, we see the effect of siRNA-mediated knockdown of PCM1. So they're knocking down PCM1 in these various conditions and basically seeing what it does. And when PCM1 is knocked down this RF is these are control conditions. It's basically a scrambled siRNA. When PCM1 is knocked down relative to control conditions, we see increased WIPI staining. So when PCM1 is knocked down, we see more WIPI staining. So we see more autophagosome biogenesis. We see more GABARAP. So when you knock down PCM1, it's almost as if GABARAP is being released. And we don't see difference in LC3. We see a slight increase in P62. So it might sound paradoxical at first that you see increased autophagy when PCM1 is knocked down, but the accumulation of these various proteins such as WIPI and GABARAP suggests that autophagy is actually slowing down because this cellular congestion caused by EB is basically like a traffic jam, and it causes things to accumulate. And we see things accumulating more quickly when PCM1 is knocked down. So GABARAP is not being trafficked, so the turnover of autophagic compartments is slowing down, and they're accumulating when PCM1 is knocked out. So we actually see PCM1 having a critical function in promoting autophagy, potentially or probably through its interaction with GABARAP. Well, it turns out that PCM1 is not just binding and moving with GABARAP, but it's actually promoting its stability. So in the bottom left graph down here, we're seeing that the overexpression of PCM1 GFP, so they're overexpressing PCM1 in these conditions. We see increased P62. We see, at the bottom, move my cursor up here, we see increased GABARAP. So GABARAP is actually increasing when you increase PCM1. So this suggests that the presence of PCM1 is somehow stabilizing GABARAP and preventing its entrance into autophagy. And on the bottom right, we see that on the other side of the coin, that the complete loss of PCM1 causes GABARAP to deteriorate following induction of autophagy. So we see, they use this cyclohexamide molecule, which inhibits translation and causes autophagy induction. And we see that following cyclohexamide, that GABARAP is used more quickly when PCM1 is knocked out. So PCM1 is stabilizing GABARAP, preventing its degradation without PCM1 is being degraded more quickly. And on the right, we see a couple of Western blots that are showing that PCM1 knockout using siRNA reduces GABARAP concentration. So we see in this PCM1 knockdown condition that the amount of GABARAP decreases significantly. And we see the same thing in these cells looking at GABARAP1, which is unprocessed. So the loss of PCM1 destabilizes GABARAP, leading to its degradation. Now that we've seen that PCM1 downregulation leads to GABARAP instability, the authors next want to understand what process mediates GABARAP degradation. And naturally, they wanted to start with the pericentriolar resident ubiquitin ligase, and that is MIB1, which again is known to be found at the pericentriolar material as well as at satellites. And indeed, they found that when MIB1 was upregulated, they saw, so they're upregulating it in this graph right here, they saw a 50% decline in GABARAP levels. So when you upregulate MIB1, you see half the amount of GABARAP. And they also observed that immunoprecipitation of GABARAP. So in this one, they're IP, they're pulling down GABARAP, and they're seeing staining of MIB1. So this is suggesting an interaction. And the authors did about 10 other experiments that really were probing this interaction. But for our purposes, we can simply say that following PCM1 loss, GABARAP is likely polyubiquitinated by MIB1 and degraded by the proteasome. So in this paper, we've seen evidence that GABARAP interacts with pericentriolar protein PCM1 through its Lear domain. And this GABARAP PCM1 complex is delivered to sites of autophagosome biogenesis as centriolar satellites. The loss of PCM1 upregulates GABARAP mediated autophagy, but also destabilizes GABARAP, suggesting PCM1 is critical to the sequestering of GABARAP during basal conditions. Now, the big finding, in my opinion, is that PCM1 regulates GABARAP. And since PCM1 is known to be a pericentriolar protein, we can say that GABARAP-associated autophagy is at least partially regulated by the pericentriolar satellites. And I think that by itself is very fascinating and warrants additional research. So thanks for watching, and I hope you enjoyed the video. And if you have any questions, please comment below.